Okay. So we were talking about transcription. And transcription is when the information of the DNA gets copied into the RNA, more particularly the mRNA. So let's continue. Why both the strands are not copied during transcription has the simple answer. First, if both strands act as a template, they would code for RNA molecule with different sequences. Remember, complementarity does not mean that they will be identical. And in turn, if they code for proteins, the sequence of amino acid is in the proteins would be different. Hence, one segment of DNA would be coding for two different types of protein, and this would complicate the genetic transfer or information transfer machinery. Second, two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. So that is why both the strands do not undergo the process of transcription, only one does. Now talking about the transcription unit. Now a transcription unit in DNA is defined primarily by three regions in the DNA: promoter, structural gene, and terminator. These three make up the transcriptional unit. Now, there is a convention in defining two strands of the DNA in the structural gene of transcription unit. Since two strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5' to 3'. The strand that has the polarity 3' to 5', this one acts like a template. So the template strand is the one that has the polarity from 3' to 5'. The other strand, which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription. Strangely, this strand, which does not code for anything, is referred to as the coding strand. So the 5:3 strand is known as the coding strand, but the thing to remember here is that it does not code for anything. Now, all the reference point while defining a transcription unit is made with the coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. So, first will be the template strand, and adjacent to it will be the coding strand. But remember that do not go with the name. The coding strand does not code for anything. So now here there will be the presence of the promoter. This is the structural gene, the template strand, and then there will be the terminator. Now the promoter and terminator flank the structural gene in a transcription unit. Flank means they are present on the either side or either end. The promoter is said to be located towards the 5' end, that is upstream of the structural gene. The reference is made with the polarity of coding strand. It is a DNA sequence that provides binding site for RNA polymerase. Which one? The promoter. And it is the presence of a promoter in a transcription unit that also defines the template and the coding strand. By switching its position with terminator, the definition of coding and template strand could be reversed. Now, if you take the terminator, put it this side, take the promoter, put it onto the opposite side, then the whole entire polarity will be reversed, and that will also work. Now, the terminator is located towards the 3' end, which is downstream of the coding strand, and it usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties shall be discussed while dealing with the regulation of gene expression. The transcription unit and the gene. Now, gene is the functional unit of inheritance. It is the one that gets inherited. Now, though there is no ambiguity that the genes are located in the DNA, it is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for polypeptide, the structural gene in a transcription unit could be said as monocistronic. So the type of gene, that is structural gene in the case of eukaryotes, is monocistronic, and it is polycistronic in the case of the prokaryotes or mostly in bacteria. Now, in eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequence or express sequence are defined as exons. These are the ones that code actually. Now, the exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Now, introns are intervening sequences, like they are present in the middle. Now, they do not appear in the mature or processed RNA. Why? Because they are removed later on. The split gene arrangement further complicates the definition of gene in terms of a DNA segment. But just remember that the DNA contains segments that are known as cistron, and it is monocistronic in the case of eukaryotes and polycistronic in the case of prokaryotes. Now, inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. Now, talking about the types of RNA and the process of transcription. In bacteria, there are three major types of RNA, that is mRNA, messenger RNA, tRNA, transfer RNA, and ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, the tRNA brings amino acid and reads the genetic code, and the rRNA plays the structural and catalytic role during translation, that is protein formation. So all the three different types of RNAs, they have some function to perform, and that is what they do. Each of them have their own unique functions to perform. Now moving forward, there is single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to the promoter and initiates the transcription. So the first process or the first step of transcription is initiation. And when does it happen? When the promoter and the RNA polymerase, first of all, the RNA polymerase comes and binds with the promoter. Now, it uses nucleoside triphosphates as substrate and polymerizes in a template-dependent fashion, following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation. So the second step, like so that it goes throughout the length of the DNA, that is the elongation. Now, only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase. This results in the termination of transcription. So this is how transcription happens. The three major steps in transcription is initiation, elongation, and termination. Now, an intriguing question is that how is the RNA polymerase able to catalyze all the three steps, which are initiation, elongation, and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with the initiation factor, that is sigma, and the termination factor, that is rho, to initiate and terminate the transcription respectively. So here we are seeing the sigma factor is the initiation factor, and then the rho factor is the termination factor, because of which initiation happens and also the termination can take place. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, that is the cytoplasm, because they do not have a nucleus, many times translation can begin much before the mRNA is fully transcribed. So in the case of eukaryotes, there will be you can say, additional steps that happen after the transcription is done, but not in the case of prokaryotes. Now in eukaryotes, there are two additional complexities. There are at least 3 RNA polymerase in the nucleus. In addition to the RNA polymerase found in the organelles, there is a clear-cut division of labor, meaning each of them have their own function to perform, such as the RNA polymerase 1. It transcribes rRNA, that is ribosomal RNA, under which comes 28S, 18S, and 5.8S. Whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5srRNA, and snRNA, that is small nuclear RNA. Here SR is small ribosomal RNA. The RNA polymerase 2 transcribes the precursor of mRNA, that is the hnRNA, that is heterogeneous nuclear RNA. So we are seeing that even though there is the presence of 3 different types of RNA polymerases, but each of those have their own function to perform. There is a clear-cut division of labor. Now, the second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Hence, it is subjected to a process called as splicing. So, after the mRNA is synthesized, it contains the exons also, it contains the introns also. Because of which, the additional step, that is splicing, has to happen so that the introns could be removed because they are non-coding stuff, right? They are not needed. So they are removed and exons are joined in a defined order. The heterogeneous nuclear RNA and undergoes additional process known as capping and tailing. Only happens in hnRNA. In capping, an unusual nucleotide, that is methyl guanosine triphosphate, is added to the 5' end of the hnRNA. In tailing, adenylate residues, like 200 to 300 adenylate residues, are added at the 3' end in a template independent manner. It is the fully processed hnRNA, now called as the mRNA, that is transported out of the nucleus for translation process. So initially, like before the formation of actual mRNA, the hnRNA is formed, that gets sort of, you can say, modifications, because of which now it moves on and forms the mRNA ultimately. Now, the significance of such complexities is now beginning to be understood. The split gene arrangements represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of the RNA world. Many a times this can be asked in the question, like this has been asked, like what represents the dominance of the RNA world? Now, in recent times, the understanding of RNA and RNA-dependent processes in the living system has assumed more importance. So this was about the transcription. Now tell me if you have any doubt.
No, ma'am.
No, ma'am.
Now talking about the genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides, that is nucleic acid, to synthesize the polymer of amino acid, that is polymer of amino acid is yes, protein. Neither does any complementarity exist between nucleotides and amino acid, nor could any be drawn theoretically. There existed ample evidences, though, to support the notion that change in the nucleic acid, that is the genetic material, were responsible for change in amino acids in the protein. This led to the proposition of a genetic code that could direct the sequence of amino acid during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and the deciphering of the genetic code were most challenging. Deciphering means decoding it. In a very true sense, it required involvement of scientists from several disciplines, such as the physicists, organic chemists, the biochemists, and the geneticists. It was George Gamow, who was a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of those bases only. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was very bold proposition because a permutation combination of 4 to the power 3, that is 4 cube, that is 4 into 4 into 4, could generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. Like each of those codon was made up of 3 nitrogenous bases. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases, that is homopolymers and copolymers. Then Marshall Nierenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo-Ochoa enzyme, polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template-independent manner. Enzymatic synthesis of RNA. Finally, a checkered board for genetic code was prepared. This is the checkered board for genetic code. So there are 64 codons and they are triplets. They are in the form of a triplet. Now, the very important part, that is the salient features of genetic code are following. The codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acid. Hence, they function as to stop codons. Some amino acids are coded by more than 1 codon. Hence, the code is degenerate. It doesn't mean that 1 codon is for 1 amino acid. There are many that can code for 2. The codon is read in mRNA in contiguous fashion. There are no punctuation, meaning it is read continuously. The triplet is read as a triplet. There is no punctuation in the middle, no stopping. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine, that is an amino acid. Some exception to this rule have been found in mitochondrial codons and in some protozoans. So even though the code is universal, there are some exceptions found. AUG, it has dual function. It codes for methionine and it also acts like an initiator codon. Lastly, UAA, UAG and UGA, these are the stop terminator codons. They do not code for anything, but wherever they are present in any sequence, they will stop the process of translation. So these are the salient features. They are very important to remember. Now moving forward. Mutations and genetic code. The relationships between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 4. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutation will be explained here. A classical example of point mutation will be is a change of single base pair in the gene for beta globin chain that results in the change of amino acid residue glutamate to valine. This happens in the disease that is known as sickle cell anemia. Here, substitution happens. If you have studied the principle of inheritance, you would know about this disease, right? Now, effect of point mutation that inserts or deletes a base can be understood by this example. Ram has red cap. Okay, so this example shows that because of the presence of a different kind of amino acid or a codon anywhere, it can cause the whole entire reading to change. Or as far as in the case of sickle cell anemia goes, from the glutamate that was originally supposed to be there, if that gets, you can say, substituted by valine, then that causes a disease. So the insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutation. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onward. But if one codon is changed and not deleted anywhere else, so the reading frame shifts. So that is a frame shift mutation. Now talking about the tRNA. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link it to amino acids, because amino acids have no structural specialties to read the code uniquely. He postulated the presence of another adapter molecule that would on one hand read the code and on the other hand would bind to specific amino acid. The tRNA, then called as the sRNA, that is soluble RNA, was known before the genetic code was postulated. So it has dual functions. Apart from reading the codon, it would also bind with the specific amino acid. Now the tRNA, it has an anticodon loop that has bases complementary to the code, and it has an amino acid acceptor end to which the amino acid binds. tRNAs are specific for each amino acid. Now for initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNAs for stop codon. Now in the figure 5.12 is the secondary structure of tRNA that has been depicted to look like a clover leaf. In actual structure, the tRNA is a compact molecule which looks like an inverted L. But this figure that is right there on your screen is the, you can say, secondary structure. It is the 2D structure of a tRNA molecule. Now talking about translation. Now translation is the protein synthesis. Now, translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are joined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as a peptide bond. Formation of a peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process which is known as charging of tRNA or amino acylation of tRNA, to be more specific. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation because catalysts obviously increase the rate of reaction. Now, the cellular factory responsible for synthesizing protein is the ribosome, because we know that ribosomes are protein factories. Now, the ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst, that is 23S rRNA in bacteria is the enzyme that is known as ribozyme for the formation of peptide bond. So it, apart from synthesizing the proteins, it also acts as the enzyme, and that enzyme is ribozyme. Now, a translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. And mRNA also has some additional sequences that are not translated and are referred to as the untranslated regions, UTR. UTRs are present at both the 5' end before the start codon and at the 3' end after the stop codon. They are required for efficient translation process. Now, for initiation, the ribosome binds to the mRNA at the start codon, AUG, that is recognized only by the initiator tRNA. The ribosome proceeds to elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. The amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. So the whole entire protein gets released, and this is how the process of translation takes place. Now talking about regulation of gene expression. Now, regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at the transcriptional level, processing level, transport of mRNA from nucleus to the cytoplasm, and then the translation level. So this is for the eukaryotes. Like in all these places, the regulation of gene expression can happen. Now, genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called as the beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide, that is lactose, into galactose and glucose, meaning to break down lactose into glucose and galactose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme, that is beta-galactosidase. Therefore, in simple terms, it is the metabolic, physiological or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also a result of coordinated regulation of expression at several sets of genes. In prokaryotes, the control of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize start site. These regulatory proteins can act both positively, such as the activators, and negatively, such as the repressors. We are talking about how gene expression can take place at the prokaryotic level. Now, the accessibility of promoter regions of the prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed as operator. The operator region is adjacent to the promoter elements in most operons, and in most cases, the sequences of the operator bind a repressor protein. Each operon has its specific operator and the specific repressor. For example, the LAC operator is present in the LAC operon. LAC refers to the lactose, and it interacts specifically with LAC repressor only. Now talking about the LAC operon. So the elucidation of the LAC operon was also a result of a close association between a geneticist, that is Francois Jacob, and a biochemist, Jack Monod. Jacob and Monod. Okay, they are responsible for the elucidation of the LAC operon. They were the first to elucidate a transcriptionally regulated system. In LAC operon, LAC refers to lactose. A polycistronic structural gene is regulated by a common promoter and regulatory gene. Such arrangement is very common in bacteria and it is referred to as operon. To name a few examples, there is lac operon, tryptophan operon, then ARA operon, histidine operon, valine operon. So all of these are the different kinds of operons that happen to have been existing in the case of bacteria. Now, the lac operon consists of one regulatory gene, that is the I gene. Here, the term I does not refer to inducer. Rather, it is derived from the word inhibitor. Apart from that, there are three structural genes, that is Z, Y, and A. Now, the I gene codes for the repressor of the lac operon. The Z gene codes for beta-galactosidase, which is primarily responsible for hydrolysis of the disaccharide, that is glucose, into its monomeric units, that is galactose and glucose. So the lactose, when it gets broken because of the enzyme beta-galactosidase that is synthesized or coded by the Z gene. The Y gene codes for the permease, which increases permeability of the cell to beta-galactoside. The A gene encodes for transacetidase. Hence, all the three genes products in the lac operon are required for metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. This is how this thing can be, asked in a diagrammatic view, and maybe you will be asked to label which part is what. So here, the repressor, when it will bind to the operator region, so then in that time, the you can say gene expression will not happen. But if the repressor gets in, like the inducer gets to the repressor and it blocks the repressor, then the whole thing will be carried forward. Now, lactose is the substrate for the enzyme beta-galactosidase, and it regulates switching on and off of the operon. Hence, it is termed as the inducer. So, lactose is the inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease enzyme. Remember, a very low level of expression of lac operon has to be present in the cell at that time. Otherwise, lactose cannot enter the cell. The lactose then induces the operon in the following manner. So when lactose is present, then what happens? The repressor of the operon is synthesized all the time constitutively from the I gene. So the I gene makes the repressor. The repressor protein binds to the operator region of the operon and prevents the RNA polymerase from transcribing the operon. In the presence of an inducer, such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows the RNA polymerase access to the promoter and transcription proceeds in that. So essentially, the regulation of lac operon can also be visualized as the regulation of enzyme synthesis by its substrate. So when lactose will be present, the whole entire process of the lac operon will take place. Remember, the glucose or galactose cannot act as inducers for the lac operon because it is the lac operon that is the lactose operon, and it runs because of the lactose, which is the disaccharide. Now, the regulation of lac operon by repressor is referred to as negative regulation. Why? Because the lactose is actually binding with that to get it inactivated. Lactose is under control of positive regulation as well, but it is beyond the scope of discussion. Good. So with this, we have completed the the lac operon. Now tell me if you have any doubt.
No, ma'am.
Everything is clear up till now?
Yes, ma'am.
Okay, now we'll talk about the Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then the DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequence, a very ambitious project of sequencing human genome was launched in the year 1990. So a question can be asked, like, in which year the Human Genome Project was launched. The Human Genome Project, HGP, was called as a mega project. You can imagine the magnitude and the requirements for the project if we simply define the aims as the follows. Now, human genome is said to have approximately 3 into 10 to the power 9 base pairs, and if the cost of sequencing required is the US $3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately 9 billion US dollars. Further, if the obtained sequence were to be stored in typed form in books, then each page of the book contained 1,000 letters and each book contained 1,000 pages. Then 3,300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of the new area that is known as bioinformatics. So, Human Genome Project comes under the bioinformatics. What are the goals of this project? Identify all the approximately 20,000 to 25,000 genes in human DNA, determining the sequence of 3 billion chemical base pairs that make up the human DNA. Store this information in databases, improve tools for data analysis, transfer related technologies to the sectors such as industries, address the ethical, legal, and social issues that may arise from the project. Okay, ethical, legal, social issues, LC, that may come from the project. Now, the Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China, and others. The project was completed in the year 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent thousands of disorders that affect human beings. Because in this way, we are actually knowing about each of the genome that is there in the human DNA, right? Each of the genes that are found. And if that is being found, then 100% it would, you can say, revolutionize the whole entire healthcare system, because by that, the diseases can be treated in a much better way. Besides providing clues to understand human biology, learning about non-human organisms, DNA sequence can lead to an understanding of their natural capabilities that can be applied towards solving challenges in the healthcare, agriculture, energy production, environmental remediation. Many non-human models, such as the bacteria, yeast, Caenorhabditis elegans, that is a free-living, non-pathogenic nematoda. Drosophila, that is the fruit fly plants such as the right and Arabidopsis, and these also, like all of these that are mentioned, these are the non-human models that have their genes sequenced or have their DNA sequenced. So you should know the examples here. Now the methodologies. The method involves two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as expressed sequence tag, EST. The other took the blind approach of simply sequencing the whole set of genomes that contain all the coding and non-coding sequence, and later assigning different regions in the sequence with functions, a term referred to as sequence annotation. So two of the things are taken for this process. Now for sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Recall that DNA is a very long polymer and there are technical limitations in sequencing very long pieces of DNA. So that is why smaller pieces of it were made. After that, cloning was done in a suitable host using specialized vectors. Cloning means what? Making the number into, like you can say, multiples of the number. Cloning resulted into, yes, amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were known as the bacterial artificial chromosome and the yeast artificial chromosome. The fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing methods for determination of amino acid sequences in protein. Now, these sequences were then arranged based on some overlapping regions present. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed for the whole entire alignment of the sequences that were found. Now, the sequence were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes, and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonucleases, recognition sites, and some repetitive DNA sequences that are known as microsatellites. One of the applications of polymorphism in repetitive DNA sequence shall be explained in the next section, that is DNA fingerprinting. So this was all about the methodology of Human Genome Project. Now talking about the features of the human genome that was found out later on. The human genome contains 3164.7 million base pairs. The average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. So dystrophin has these many bases and it is the largest known human gene. The total number of genes is estimated at 30,000, much lower than the previous estimates of 80,000 to 140,000 genes. Almost all, that is 99.9% nucleotide base pairs are exactly the same in all the people. The functions are unknown for 50% of the discovered genes. So many of the genes we don't know the functions of. Less than 2% of the genome codes for proteins. Repeated sequences make up very large portion of human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has most genes, that is 2,968, and the chromosome Y has the fewest gene, that is 231. Scientists have identified about 1.4 million locations where single base differences, the single nucleotide polymorphism, that is also pronounced as SNPs, occur in humans. So in these many locations, 1.4 million locations is where the SNPs occur. This information promises to revolutionize the process of finding chromosomal locations for disease-associated sequences and tracing human history. Now, applications and future challenges. Deriving meaningful knowledge from DNA sequences will define research through coming decades, leading to our understanding of biological systems. The enormous task will require the expertise and the creativity of tens of thousands of scientists from varied disciplines in both the public and the private sectors worldwide. One of the greatest impacts of having the human genome sequence may well be enabling a radically new approach to the biological research. In the past, researchers studied one or few genes at a time. With whole genome sequences and a new high throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or an organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. So all of these, like, because of the human genome sequencing, because we know now how many base pairs are present in what kind of genes, there are multiple things that can be benefited because of that. Any doubts up until now?
No, ma'am.
No, ma'am.
Now we'll move on to the DNA fingerprinting. 99.9% of base sequence among humans, every single human, is same. Assuming human genome has 3 into 10 to the power 9 base pairs. So basically, all of the humans that exist on the earth, 99.9% of their sequence is same. But there is a difference, a little difference is there. Now, it is these differences in the sequence of DNA which make every individual unique in their phenotypic appearance. Phenotypic is the morphological or external appearance. Now, if one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 6 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. Now, DNA fingerprinting, it involves identifying differences in some specific regions in DNA sequence that is known as the repetitive DNA. Because in these sequences, a small stretch of DNA is repeated many times. These repetitive DNA are separated from the bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred to as satellite DNA. Depending on the base composition, that is adenine to thymine richness or guanine to cytosine richness, the length of segment and the number of repetitive units, the satellite DNA is classified into many categories, such as microsatellites, mini satellites, depending upon the ratio of the nitrogenous bases, the length of the DNA, and the number of repetitive units. These sequences normally do not code for any protein, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue, such as the blood, hair follicle, skin, bone, saliva, sperm, from an individual show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphisms are inheritable from parent to children, DNA fingerprinting is the basis of paternity testing in the case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism, variation at genetic level, arises due to mutation. New mutations may arise in individual either in somatic cells that are the body cells or in the germ cells that are the sex cells. If a germ cell mutation does not seriously impair individuals' ability to have offspring who can transmit the mutation, then it can spread to the other members of the population through the sexual reproduction if it is not detrimental. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant or more than one allele at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA would be higher, as mutation in these sequences may not have any immediate effect or impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of variability or polymorphism. There is a variety of different types of polymorphisms, ranging from single nucleotide chain to very large scale changes. For evolution and speciation, such polymorphisms play a very important role, and you will study these at higher classes. The technique of DNA fingerprinting was developed by Alec Jeffreys. Important. He used a satellite DNA as probe that shows a very high degree of polymorphism. It was called as VNTR, that is variable number of tandem repeats. The technique, as used earlier, involves southern blot hybridization using radio-labeled VNTR as a probe. It included, now what are the steps of DNA fingerprinting? Isolation of DNA, digestion of DNA by restriction endonucleases, separation of DNA fragments by gel electrodes, then transferring or blotting of separated DNA fragments to synthetic membranes such as nitrocellulose membrane or nylon membrane, then hybridization using labeled VNTR probe, and lastly detection of hybridized DNA fragments by the process of autoradiography. The VNTR belongs to a class of satellite DNA, that is the mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The number of repeats show very high degree of polymorphism. As a result, the size of the VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for individual DNA. It differs from individual to individual in a population except in the case of monozygotic twins, that are the identical twins. Now, the sensitivity of the technique has been increased by the use of polymerase chain reaction, that is for amplification. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider application, such as in determining population and genetic diversity. Currently, different probes are used to generate DNA fingerprints. So, the DNA fingerprinting, apart from being used in the forensic department, it is also used for these two other things, that is determining population diversity and genetic diversity. With that, we have completed the chapter. Now tell me if you have any doubt. Everything is clear, students?
Yes, ma'am.
The DNA fingerprinting, translation, every topic is clear, right?
Yes, ma'am.
Okay, good. Now, which chapter should I start after this? Any chapter that you need help in from class 12?
Let's go with evolution.
Hmm?
Let's go with evolution.
Evolution.
Yes, ma'am.
Okay. Okay, just take 5 minutes. Okay, go through the NCERT chapter, this one, the molecular basis. Okay, just take 5 minutes and then we'll start evolution. Okay. Yes, students, any doubts?
No, ma'am.
Okay. Now we'll start with evolution, the next chapter. Okay, evolutionary biology is the study of history of life forms on Earth, how the life forms originated and how the things happen. What exactly is evolution? To understand the changes in flora and fauna that have occurred millions of years on Earth, we must have an understanding of the context of origin of life, that is the evolution of Earth, of stars, and indeed the universe itself. What follows is the longest of all the construed and conjectured stories. This is the story of origin of life and the evolution of life forms or biodiversity on planet Earth in the context of evolution of Earth and against the background of evolution of universe itself, talking about the origin of life. When we look at the stars on a clear night sky, we are in way looking back in time. Stellar distances are measured in light. What we see today is an object whose emitted light started its journey millions of years back and from trillions of kilometers away and reaching our eyes now. However, when we see objects in our immediate surrounding, we see them instantly and hence in the present time. Therefore, when we see stars, we apparently are stepping into the past. The origin of life is considered a unique event in the history of universe. The universe is vast. Relatively speaking, the Earth itself is almost only a speck. The universe is very old, that is almost 20 billion years old. Huge clusters of galaxies comprise the universe. Galaxies contain the stars, the clouds of gas, and dust. Considering the size of universe, Earth itself indeed is a speck. Now, the Big Bang Theory attempts to explain to us the origin of universe. It talks of a singular huge explosion unimaginable in terms. The universe expanded and hence the temperature came down. Hydrogen and helium formed sometime later. The gases condensed under gravitation and formed the galaxies of the present day universe. In the solar system of the Milky Way galaxy, Earth was supposed to have been formed about 4.5 billion years back. There was no atmosphere on the early Earth. Water vapor, methane, carbon dioxide, and ammonia released from molten mass covered the surface. The UV rays, the ultraviolet rays from the sun, broke up water into hydrogen and oxygen, and the lighter, that is the hydrogen, escaped. It went back into the atmosphere. Now, oxygen combined with ammonia and methane to form water, CO2, and others. The ozone layer was formed at that time. As it cooled, the water vapor fell as rain to fill all the depressions and form the oceans. Life appeared 500 million years after the formation of Earth. That is almost 4 billion years back. Did life come from outer space? Some scientists believe that it came from the outside. Early Greek thinkers thought units of life called as spores were transferred from different planets, including Earth. Panspermia is still a favorite idea for some astronomers. They thought that life was existing somewhere else, and when the Earth was formed, then the life was transferred here in the form of spores. That is the unit of life. For a long time, it was believed that life came out of decaying and rotting matter such as straw, mud, etc. This was the theory of spontaneous generation. The theory of panspermia was the first theory. Then there is the theory of spontaneous generation. Louis Pasteur, by careful experimentation, demonstrated that life only comes from pre-existing life. He showed that in pre-sterilized flask, life did not come from killed yeast, while in another flask open to air, new living organism arose from the killed yeast. Spontaneous generation theory was dismissed once and for all. However, this did not answer how life came on Earth. Now, Oparin of Russia and the Oparin of Russia and Haldane of England proposed that the first form of life could have come from pre-existing non-living organic molecule, meaning some of the non-living molecule like the biomolecules such as the RNA protein. And that formation of life was preceded by chemical evolution. So long before biological evolution took place, chemical evolution was there. This was something that Oparin and Haldane explained. That is formation of diverse organic molecules from inorganic constituents. The conditions on Earth were high temperature, volcanic storms, reducing atmosphere which contained CH4, NH3, etc. Now, in the year 1953, S.L. Miller, an American scientist, created similar conditions in a laboratory scale. He created electric discharge in a closed flask containing CH4, H2, and NH3 and water vapor at 800 degrees Celsius. He observed formation of amino acid. In similar experiments, others observed formation of sugar, nitrogen bases, pigments, and fats. Analysis of meteorite content also revealed similar compounds, indicating that similar processes are occurring elsewhere in the space, meaning on different planets as well. So with this limited evidence, the first part of the conjectured story, that is the chemical evolution, was more or less accepted. We have no idea how the first self-replicating metabolic capsule of life arose. The first non-cellular form of life could have originated 3 billion years back. They would have been giant molecules such as RNA, proteins, and polysaccharides. All of these are non-cellular because these are non-living. Now, these capsules reproduce their molecules. Perhaps the first cellular form of life did not possibly originate till about 2000 million years ago. These were probably single celled. All life forms were in water environment only. So initially the whole entire, you can say, ecosystem or the place was water filled. This version of biogenesis, that is the first form of life, arose slowly through evolutionary forces from non-living molecule, is accepted by majority, chemical evolution. However, once formed, how the first cellular forms of life would have evolved into complex biodiversity is again unknown thing. Now, evolution of life forms, that is a theory. Now, a conventional religious literature tells us about the theory of special creation. This theory has three connotations. One, that all living organism species that we see today were created as such. Two, that the diversity was always the same since creation and will be the same in the future also. Three, that the earth is about 4,000 years old. All these ideas were strongly challenged during the 19th century based on the observations made during a sea voyage in a sail ship called as HMS Beagle around the world. Charles Darwin concluded that existing living forms share similarities to varying degrees, not only among themselves, but also with them life that existed millions of years ago. There is some kind of, you can say, similarity, or they shared the certain appearance, the certain structures that were there. Many life forms of those do not exist anymore. There had been extinctions of different life forms in the years gone by as new forms of life arose at different periods of history of Earth. There has been gradual evolution of life forms. Any population has built-in variation in characteristics. What is variation, girls? Variation? What is variation? No one knows variation.
Ma'am, can you repeat what you just asked?
The question is, what is variation?
Differences.
Differences as in? Variation is the introduction of new characters. Some way or the other, the population has new character introduction in them, which makes them different from the previous. Those characters which enable some to survive better in natural conditions, that is climate, food, physical factors, etc., would outbreed others that are less endowed to survive under such natural conditions. Another word uses is the fitness of the individual or the population. Now, the fitness, according to Darwin, refers only to the reproductive fitness. Hence, those who are better fit in an environment leave more progeny than others. These, therefore, will survive more and hence are selected by the nature. He called it natural selection and implied that it was as a mechanism of evolution. So, one of the mechanism of evolution is natural selection. That was explained by Darwin. Now, let us also remember that Alfred Wallace, a naturalist who worked in Malay archipelago, he had also come to the similar conclusion at the same time. In due course of time, apparently new types of organisms are recognizable. All the existing life forms share similarities and share common ancestors. However, these ancestors were present at different periods in the history of Earth, such as different epochs, different periods, and different eras. The geological history of Earth closely correlates with the biological history of the Earth. A common permissible conclusion is that the Earth is very old, not thousands of years as was thought earlier, but billions of years old. Now the Earth. Now, what are the evidences for evolution? How do we know that the evolution did actually take place? Evidence that evolution of life forms has indeed taken place on Earth has come from many quarters. Fossils are remains of hard parts of the life forms found in the rocks. Rocks form sediments, and a cross section of Earth's crust indicates the arrangement of sediments one over the other during the long history of Earth. Different age rock sediments contain fossils of different life forms who probably died during the formation of the particular segment. Some of them appear similar to modern organisms. They represent extinct organisms, that is, the dinosaurs. A study of fossils in different sedimentary layers indicates the geological period in which they existed. So, because of the presence of fossil, we can understand that, okay, a certain life form existed a long time back, and we can actually find it out by looking at the soil and the way the sediment is. The studies show that the life forms varied over time, and certain life forms are restricted to certain geological time spans. Hence, new forms of life have arisen at different times in the history of Earth. All this is called as paleontological evidence. Paleontology is the study of fossils, right? So that is the paleontological evidence. Now, comparative anatomy and morphology shows similarities and differences among the organisms of today and those that existed years ago. Such similarities can be interpreted to understand whether common ancestors were shared or not. For example, whales, bats, cheetah, and who, all of them are mammals, share similarity in the pattern of their bones of 4 limbs. So their upper limbs, they have a similarity of pattern of bones. Though these 4 limbs perform different functions in these animals, they have similar structure, similar anatomy is there. All of them have the humerus, radius, ulna, carpals, metacarpals, and phalanges in their 4 limbs. Hence, in these animals, the same structure developed during different directions due to adaptations to different needs. So, this type of evolution is divergent evolution, wherein the structure is same, but the function is different. And these structures are homologous structures. Homology indicates common ancestry. Other examples are vertebrate hearts or brains. In plants also, the thorn and the tendrils of Bougainvillea and Cucurbita represent homology. So the examples of homology is like these examples, they're very important. Now, homology is based on divergent evolution, whereas analogy refers to the situation exactly opposite, where they do not have a similar structure, but they have a similar function. The wings of butterfly and the wings of bird, they look alike, but they are not similar. But they have a similar function, that is to fly. So these are the analogous structures, and this type of evolution is convergent evolution. Now, out of these two evolutions, the one that denotes a common ancestry is the divergent evolution. Other examples of analogy are the eye of octopus and the eye of mammals. Both made up of different things, but perform the same function, that is the sight. Flippers of penguins and dolphins, both made up of different things, but again perform the same function of swimming. One can say that it is the similar habitat that has resulted in selection of similar adaptive features in different groups of organisms, but toward the same function. Now, sweet potato, which is a root modification, and potato, which is a stem modification, this is another example of analogy. Now, in the same line of argument, similarities in proteins and genes performing a given function among diverse gives to common ancestry. These biochemical similarities point to the same shared ancestry as structural similarities among diverse organisms. So, convergent and divergent evolution, extremely important to remember, extremely important. Now, man has bred selected plants and animals for agriculture, horticulture, sport, or security. Man has domesticated many wild animals and crops. This intensive breeding program has created breeds that differ from other breeds, such as dogs, but still are of the same group. It is argued that if within hundreds of years, man could create new breeds, could not nature have done the same over millions of years? So if man can create new breeds, then the nature could have created multiple new breeds. Any doubts up until now? You've understood convergent and divergent evolution?
Yes, ma'am.
All right, so we'll keep it till here only for today. Do you have any doubts in this chapter up until now? You've understood the different theories of evolution that came around initially?
Yes, ma'am.
All right. Okay, so with that being said, let's just keep it till here only. And in the next class, we'll continue with more of the evolution for evidences and evidences for evolution, and then what is adaptive radiation and all of the different. Up until now, do you people have any doubts?
No, ma'am.